A colleague from Imperial, Ruggero Cortini. Thanks very much.
Thank you. Today I'll be talking about chiral effects in DNA supercoiling. The first thing I want to mention is my acknowledgements to my supervisor, of course, Alexei Kornishev, and to the people in the research group there. Also from Imperial, from the labs of Jeff Baldwin and Tim Albrecht, and from our collaborators at the University College of London in the Mathematics Department, and other collaborators as well from various other groups, as Lindsay Drich, who is in the audience right now, and Graham King from Vrije Universiteit Amsterdam, who's also in the audience right now. So in my talk I want to tell you about what happens if you take the Kornishev-Lakin theory of DNA-DNA interactions and you apply it to the case of DNA supercoiling. Turns out that there are very important and significant effects that might arise because of it. So I'll tell you briefly about the Kornishev-Lakin theory, and then I'll tell you about what are its applications in the case of free braids of DNA and closed circular DNA. So let me start by telling you in very few words what the Kornishev-Lakin theory is. It's a theory of DNA-DNA interactions of intact double-stranded DNAs in its electrostatic interactions. So it was initially devised to explain the occurrence of DNA-DNA attraction in the presence of DNA condensers. So unlike other theories of DNA-DNA interactions, the main point is that it takes into account that the charges on the surface of DNA are on a helical pattern, follow a helical pattern. That's a fundamental thing. So the negative charges on DNA are fixed and are situated on the phosphate groups. And the main assumption is that the positive charges are also following helical lines. So, and there is a certain amount of positive charge which is located at the minor and the major grooves of the DNA. And there's a fundamental parameter in the theory which is called theta, which is the total amount of negative charge that is compensated by bound counterions at the DNA surface. Now, under these assumptions, modeling the DNA as a low dielectric core with these helical patterns of charge fixed at its surface, you calculate the electrostatic interaction energy using the Bi-Huckel equation. There's a lot of calculations to be done. Probably the right units of measure is kilograms of paper to do the calculations. There's a lot of calculations. I won't show you the formulas. And the result, the main result, is that taking into account these helical patterns, attraction between DNA molecules can occur because of a mechanism which was called the electrostatic zipper, which is essentially that on opposing surfaces, positive charges and negative charges might align to each other, and this locking of positive and negative on opposing surfaces gives rise to attractive interactions. And the parameter theta actually discriminates between repulsive interaction curves and attractive interaction curves. So when theta exceeds a certain threshold, attraction between DNA molecules can occur. So this is the first thing I wanted to tell you about. The second thing is that this mechanism of electrostatic zipper relies heavily on the fact that positive and negative charges have to be aligned and stay aligned throughout the whole length of the molecule. So any distortion from the ideal helical line in the patterns of charge also disrupts this mechanism of alignment of positive and negative charges. Now it turns out that the distortions from the ideal helical lines are sequence dependent. So this tells you that the mismatch between the alignment of positive and negative charges is accumulating for non-homologous sequences and is staying the same for homologous sequences. As a result, for For rigid molecules, what happens is, as a function of the DNA length, for identical sequences, the interaction energy continues to go down under the conditions in which attraction occurs. And at some point, unrelated sequences lose the favorable interaction energy. So the bottom line of the slide is attractive interactions between homologous DNAs are stronger than non-homologous DNAs based on this mechanism of electrostatic zipper. The final thing I want to mention in the introduction is that once you take into account the helical patterns of charge on the DNA surface, you also realize that the crossings between DNA molecules are not equivalent. So unlike theories in which the charge is assumed to be homogeneously distributed on the surface of DNA, this theory predicts that this crossing, in a so-called right-handed crossing, will be more favorable than this crossing because negative and positive charges are better aligned than in the other case. And this is because DNA is a right-handed helix, the right-handed crossing is favored. So the idea was to apply this theory to DNA supercoiling, in which these crossings occur all the time in DNA plectonemes. So this was the idea of my PhD, and so the first thing to do this, to apply this, is to calculate the electrostatic interaction not between parallel molecules anymore, but what are the electrostatic interactions between a pair of double-stranded DNAs when they are braided around each other, which is the configuration in which their distance stays constant, and also their tilt angle locally remains constant. So they take advantage both of the attractive interactions between parallel DNA and the advantage of having a local tilt. So the main ingredients are in the attraction, which is modeled in this term in the total energy E0, which was already calculated back in '97, and then the bending energy, which is an energetic cost of bending, of course, which is calculated in the elastic rod theory, and then the chiral torque. And this was the first thing that I had to calculate. The calculation was sketched by Dominic Lee and took several kilos of paper to be performed. So what I want to tell you is the results of the theory. And as I was showing before in the cartoon, left-handed braids in which these right-handed crossings are occurring are always favored over right-handed braids. And this picture here shows that if you select a particular value of theta, if this angle alpha, which is the tilt angle between the DNAs, is 0, you never have attraction. But in those conditions, if you tilt in the positive direction, which means forming a left-handed braid, you can have an attractive well. So this is the key result, because for instance, here I show the minimum energy plotted as a function of theta, and it's quite a big effect that you have because of braiding. So you can see that for parallel molecules, the attraction starts around 85% theta, and for a braid it starts at much lower values of theta, so much less charge compensation is needed for attraction to occur. So where can we find these braids? Where can we expect these phenomena? So luckily, when my PhD started, a very important paper came out of the group of Mara Prentice in Harvard, in which they detected that homology enhanced pairing in monovalent salt solution, and they detected it at a quasi-single molecule level by putting DNA tethered onto a capillary surface and the other DNAs tethered to magnetic beads, and then applying a magnetic force after the magnetic beads are added to the capillary. And once the force is applied, if you detect beads at the so-called diagnostic distance, which is approximately the contour length of DNA, that is a sign of pairing. And now this number of tethered beads increased as a function of the salt concentration, and were at equivalent salt concentrations, the number of tethered beads was higher for homologous molecules. So this was a sign that homology recognition could occur also in the absence of proteins, and it's a very important result. But also, pairing was actually not expected in monovalent salt, but because of the mechanism of braiding, which can make the attraction occur at much lower values of charge compensation, this electrostatic braiding effect might explain the occurrence of this pairing. So this is one thing. The next thing was actually to apply the theory of braiding to the case of closed circular DNA. And so we built a very, very simple model of closed loop DNA in which we take the braid that we used before, and we essentially add the two loops at the end, and take into account the torsional energy, and then the end loop bending energy, and then the braiding, the energy of the braid, and then the topological constraint which arises from closure of the DNA molecules. So the first result I want to show you is actually for a simpler case of nicked molecules, in which the closed circular DNA can adopt any conformation it would like at no torsional energy cost. And it's similar to the case of what happens for free braids. So as a function of theta, the attraction starts at a certain value, and what happens is that braiding can occur in nicked molecules. So now this theory is predicting that in the right salt environment, writhing can occur in nicked closed circular DNA. So we thought about what are the evidences for this in the literature, and we found that it's known that divalent ions wind the DNA helix. So the experiments that are performed there are that negatively supercoiled DNA are taken from cells, they are nicked, and then salt is added, like these divalent metal ions, and then they are religated and run over gels. And what happens at the end is that positive supercoiling is detected. Now this mechanism was interpreted because the twist in nicked DNA changes, and then religating them and in low salt performing gel electrophoresis, positive writhing is detected. But actually this is completely equivalent to our interpretation, which is that writhe changes when nicking occurs. So this is about nicking, but also for in the more complicated case of constrained closed circular DNA, the theory also has some striking results, which are, well, at low values of theta, the minimum energy as a function of excess linking number is the well-known parabolic shape, which tells you that you have to invest energy to add excess linking number. But since the electrostatic braiding effect actually is an energetic advantage, once the theta goes up to a sufficient amount, then actually the energy of closed loop DNAs might become negative. And the threshold for this electrostatic stabilization is starting from the positive excess linking number, because this mechanism favors positive supercoils, but also for if theta is high enough, then stabilization might occur also for negative excess linking number. So we looked back at the literature and found that there is evidence for tight supercoiling, particularly in the case of cryo-EM images taken in the group of Andrei Stasiak, and AFM images which were taken in the group of Lyubchenko. And these are the AFM images and you can see that in these images indicated by arrows, there are regions of tight interactions between distal portions of the DNA plasmid, and the extent of these close contact regions increases with increasing salt. However, it increases with negative supercoiling. So this is something that is not quite fitting with our interpretation. However, as I showed you, our model of DNA is really primitive. So many things can actually, this effect can be explained with our electrostatic braiding effect, but a much more sophisticated analysis might be needed. So in conclusion, essentially more experiments are needed to prove this, that these effects are real. In particular, 4 bead experiments that are currently underway in a group of Heijswoud in Amsterdam, single molecule experiments of which we are now preparing a theory of, and homology versus non-homology in closed loop DNA, which is work currently underway at the chemistry department at Imperial, and there are some preliminary encouraging results which I will not tell you about. And there are many other theoretical improvements, a theoretical model for these parameters, undulations of a molecular center line, structural transitions, and simulations of DNA. And thank you for your attention.
We have time for a question? Yes, Andy.
If you have these same sequence repeated twice in a supercoiled molecule, does your model predict whether you can stabilize a particular configuration of the supercoil? Does it sort of fix it by sequence pendulum interaction?
Sorry, can you say again?
Okay, if you say the sequence will enhance this type of interaction.
Yeah.
If you repeat the sequence within a long supercoiled molecule, can you then, on your model, preferentially stabilize a particular configuration of that molecule?
Well, yes. We don't have a theory for that because we have assumed that the closed loop molecules is homogeneous.
Yes.
So, but we would expect that there is that particular region in which homology occurs, that the tightening and the close contact might occur, and maybe not elsewhere. But that has to be studied.
What sort of enhancement would you expect? Or is that?
We could expect that in the regions in which the homologous sequences meet, there might be close. The two sequences might go close to each other and then be separated in the rest of the molecule. That's what I mean.
Let's thank him again for a lovely